Welcome back to another Biology of Cells and Tissues video. We're going to be continuing with the urinary system. As always, if you haven't already, drop a like on the video and subscribe if you're not yet. It really helps a lot. Okay, so we left off talking about the nephron. We talked about the renal corpuscle before, where we were doing all of the filtering. And now we're going to talk about the renal tubule, the second part of the nephron. This is where we're actually going to process the filtrate into urine. We have the proximal tubule, the loop of Henle, which has a couple other components, the distal tubule or distal convoluted tubule, and then the collecting duct. Tubule / duct, because there's both of them involved there. Starting with the proximal tubule. Well, let me see real quick first. I had a picture here. I don't remember. Here it is. I thought that I'd put this picture there as well. And this is our nephron. This up here, we're talking about where the glomerulus is. That's that renal corpuscle. Then all of the tube work coming out of it. That is what we're talking about, the tubule portion of it. Okay, so the proximal tubule, this is the first segment of the renal tubule, and its main responsibility is for a major portion of the reabsorption and the secretion processes in the kidney. So this chunk of stuff, the renal tubule, this is an entire exam in physiology 2. Just straight kidney stuff. All of this stuff that we're talking about now and then a bunch more is all covered in an entire, they do, I think, 4 weeks just on kidney stuff. So this comes back heavily, especially the tubule portion. So I really, really, really want you guys to know this and master this now, so that way you don't have to have the massive headache of relearning it later on, okay? So when we think about reabsorption and secretion, think the proximal tubule. 85% of sodium chloride and water are reabsorbed here, and then things like glucose, amino acids, and any of the other small proteins that we for sure want to keep in the body also get reabsorbed here. Except in certain pathological conditions, you'll talk about certain conditions like amino acids, for example. In biochemistry, you should have talked about a condition, or you will talk about it soon, called maple syrup urine disease. That is one where our bodies cannot break down branched chain amino acids, and so as a result, we end up with those branched chain amino acids, leucine, isoleucine, and valine inside of our urine, because essentially our body cannot break them down, so we get an excess of them built up in the blood. Or glucose in the case of diabetes. So diabetes mellitus, where we either type 1, where we don't produce any insulin, or type 2, where our cells are desensitized to insulin. We can have very high levels of blood glucose, and then we end up filtering out a ton of glucose, and that's why we have glucose in the urine in pathology. This is also kind of a weird thing, but supposedly, I'm not sure exactly how much truth there is to this, supposedly medical providers used to taste the patient's urine if they expected them having diabetes. And it would be very sweet because it's full of glucose. Again, I don't know exactly how true that is, or exactly what time period that quote-unquote fact comes from, but yeah, that's one of the things that we can remember about diabetes. Okay, so that's all that's reabsorbed. And then we secrete creatine or creatinine. We'll call it creatine. This is the waste product of metabolism, and it is secreted to be excreted or to be removed. This is something that you will eventually learn. There's a test called creatine clearance This is a lab test that they do to check the health and the function of your kidneys. If you have a good level of clearance of this, your body's clearing it appropriately, then that means the kidneys are healthy. Again, that'll come up in the future, like in lab diagnostic courses in try 4 or 5, depending on if you're blended or not. Okay, the proximal tubule is slightly longer than the distal tubule, which means it has more surface area for more absorption, and it has 2 parts. It has a convoluted or twisted part, and then it has the straight part, which goes into the loop of Henle, which we'll look at next. And of course, we want to know the epithelium of it. So it is a simple cuboid epithelium that has a brush border. Remember, we talk about the brush border. We know that we think that it has villi, and then it has microvilli, so that it looks like a brush. So that is, and we know the villi and microvilli are very, very important for increasing surface area to allow for what? More absorption, and which is the whole point of the proximal tubule. Here's the part of the proximal tubule, the convoluted part, and then the straight part. Here's what we have, those microvilli all through there. Here's what it looks like as a slice. We have the brush border right in there, capillary stuff are all around it. We have here the proximal convoluted tubule portion right in here. After the proximal convoluted tubule, we have the renal tubule. I should say after the proximal tubule, because it's the straight, runs into the loop of Henle. This is also known as the there's a picture that uses a different word. It's the sorry, let me find it real quick. Nephron loop, that's what it's called. The AKA for it is the nephron loop. Oh, it was the next slide and I went all the way down. AKA the nephron loop. So loop of Henle, nephron loop, it's the same thing. I always learned it as loop of Henle, but I guess some books will use nephron loop. So the goal of the loop of Henle, now that we've filtered out all of the fluid and all the gunk, we've reabsorbed and secreted the stuff that we want to. Now we're going to look at concentrating the urine. Kind of like when we talked about the digestive system, how the large intestine sucked all the water out of what becomes the feces. Same idea here. Essentially what we're going to do is we're going to pull out a bunch of the water and really concentrate the urine. So that way we're not losing a bunch of water to the process of urination. So a couple key points. These guys right here, these are definite testable type questions. Super easy for professors to test on these. The descending limb is permeable to water only. So no solutes, just water. The ascending limb is impermeable to water, but permeable to solutes. So in the descending limb, we're going to be pulling water out. In the ascending limb, we're going to be starting to do some fine tuning, putting in or putting out specific solutes to make sure that we're getting the right concentrations in the urine and keeping what we need. The thick ascending limb does active transport. It actively transports sodium chloride out of the tubule. We'll talk about transport types a lot more in Phys 1, and then you'll dive super deep into all of this stuff in Phys 2, that's Try 3. So here is that loop of Henle, or the nephron loop. We have the thin descending segment, the thin ascending segment, and then the thick ascending segment. Now this finally becomes the distal convoluted tubule. The distal convoluted tubule is made up of simple cuboidal epithelium with no brush border. It's smaller cells in the proximal tubule. It contains something called the macula densa, which is part of a process called the juxtaglomerular apparatus. We're going to look at that on its own in a little bit. In the distal convoluted tubule, its job is that function, that maintaining the acid-base balance. So it can secrete hydrogen or it can secrete ammonium. These are perfect test questions right here. This is to eliminate acid from the blood. If you remember acid, we think about biochemistry. Anything that is acidic, it's because the pH is low. Low pH means lots of hydrogens, so we get rid of hydrogen in order to decrease the acidity, in order to increase pH. As well, this is also where technically you'll have their opposite true, where if your body is too alkaline or too basic, then you can also secrete bicarbonate here in order to bring the pH down. So it's that fine-tuning control of maintaining our blood, part of maintaining our blood pH. Finally, the distal convoluted tubule, this is where the adrenal gland hormone aldosterone acts, and we reabsorb sodium and secrete potassium. It's kind of like an exchange. So the hormone aldosterone, which comes from the adrenal gland, also known as the suprarenal gland, it's the gland that sits on top of the kidney. It releases this hormone, and it's going to tell the distal convoluted tubule, hey, we want to keep more sodium. So it essentially does a swap. It pulls in a sodium and kicks out a potassium. This is one of the reasons why people who have high blood pressure, one of the recommendations is to increase fruit and veggie intake, increase potassium intake, or even potentially take a slight potassium supplement in order to give their bodies a better chance of kicking out sodium, or kicking out potassium, or exchanging everything, so that way there's enough potassium and enough sodium in the body. There's an equal balance of it all. Here is a view of the distal convoluted tubule. Each of these is a distal convoluted tubule. I believe it's these ones is the one that's focusing on. Now, finally, after the distal convoluted tubule, we have the collecting tubules and the ducts. These are cuboidal epithelium, and the hormone that acts here is vasopressin, also known as ADH, or antidiuretic hormone. That is released from the posterior pituitary gland up in the brain. That posterior pituitary gland sits in the sella turcica. That's that little saddle in the sphenoid bone. And its job, it increases permeability to water. So it helps it's antidiuretic. So it makes us retain and hold on to water. So then these collecting ducts, here we go. So each of these little spots here, let me draw. Pretend that, so this distal convoluted tubule comes and connects here. There's also another distal convoluted tubule connecting here, another one here, another one here, another one here. So all of these are converging. So the distal convoluted tubules all converge into the collecting duct. And then the collecting ducts, they all merge together to form what's called a papillary duct. I mentioned that word way back in part 1, and that is located at the tip of the renal pyramid, which is The renal pyramid is located in the medulla of the kidney. Those papillary ducts all drain. Papillary ducts converge and then drain and become the minor calyces. Then same thing, the minor calyces all connect to become the major calyces. Major calyces connect and become the renal pelvis. Here are these collecting tubules and ducts. Here's where they are right there. Now we're going to talk about that juxtaglomerular apparatus, which is specifically connected to our distal convoluted tubule. So the juxtaglomerular apparatus, this is a specialized structure in the kidney. Its job is to regulate blood pressure as well as help to control glomerular filtration rate. GFR, this is how much filtrate we're pulling out of the blood. Okay, and this is connected. So if we're pulling out lots of stuff from the blood, this has an effect on the blood pressure, and it can either help the blood pressure go up or down. So we're going to control all of those things. There are 4 components that we're going to talk about. We have the afferent arteriole, where the juxtaglomerular cells are located. The distal tubule, which has the macula densa. We have extraglomerular mesangial cells, also known aka Lasis cells. And then we're going to talk about the renin-angiotensin-aldosterone system, or the RAAS. So the afferent arterioles, remember, the afferent arterioles, they are bringing the blood into the renal corpuscle. They split, become the capillaries of the glomerulus, and all that kind of stuff. We have these special cells called juxtaglomerular cells. These are specialized or modified smooth muscle cells in the walls of the afferent arterioles. They detect changes in blood pressure, and they will help, and they will release an enzyme called renin whenever it is necessary, specifically when blood pressure is low. Renin, that is that enzyme. So when blood pressure in the afferent arteriole drops, the juxtaglomerular cells release renin into the bloodstream. On the other side of the equation, the distal tubule has the macula densa cells. These are specialized columnar epithelial cells that have been specialized to monitor the sodium concentration of the filtrate. So we have the afferent arterioles, the juxtaglomerular cells checking the blood pressure, the macula densa in the distal tubule monitoring the sodium concentration. If they detect that the concentration of sodium is not optimal, if the sodium levels are too low, which also could potentially correlate with low blood pressure, they're going to send a signal back to those juxtaglomerular cells in the afferent arteriole, also to release renin, which is going to eventually help restore the sodium balance and increase blood pressure. We're going to see how that happens when we get to the last step, that renin-angiotensin-aldosterone system. Mesangial cells, also known as Lasis cells, these are located between the macula densa and the juxtaglomerular cells. They provide structural support, just like the mesangial cells that we looked at before. They provide structural support, and they may also assist in the communication between the macula densa and juxtaglomerular cells for renin release. You can tell renin release is the most important part of all of this when it comes to maintaining an appropriate control of blood pressure and the sodium concentration. Okay. This is the renin-angiotensin system step by step. So renin, angiotensin, aldosterone system, the RAAS. You are going to see this so many times. So I want to make sure that we have a solid base with it now, because it's going to come back a lot. So this is a hormonal cascade that regulates blood pressure and fluid balance. So the juxtaglomerular cells, which are located in where? The afferent arterioles, monitor blood pressure. The macula densa monitors ion concentration, specifically sodium ion concentration. If either are low, they communicate with the juxtaglomerular cells to release renin. Renin is an enzyme that travels through the blood, and it acts on a protein called angiotensinogen, which is made in the liver. So it goes into the blood, it travels to the liver. There will be some angiotensinogen in the blood itself, but mostly in the liver. And renin turns angiotensinogen into angiotensin 1. This is the inactive form still of angiotensin. Next, angiotensin 1 travels to, I'm going to put this in the proper Roman numerals format because that's where you're going to see it. Angiotensin 1 travels to the lungs, and there is an enzyme called angiotensin converting enzyme known as ACE, which is located in the lung endothelial cells. And this enzyme converts angiotensin 1 into the active form angiotensin 2. Then angiotensin 2 does two things. First, it causes vasoconstriction, so it causes the blood vessels to constrict, which raises blood pressure. And then it also travels to the adrenal gland to release aldosterone, which then goes back to the distal convoluted tubule and tells it to reabsorb sodium, or helps it to reabsorb sodium. I highly recommend that you practice this loop a couple of different times. I'm focusing again from kidney releases renin from where? The juxtaglomerular cells. They'll call them JG cells. And then it acts on angiotensinogen from where? The liver. Turns it into angiotensin 1. Then it travels to the lungs where there's the ACE enzyme. Turns it into angiotensin 2. And then that goes to the adrenal cortex, releases aldosterone, and we get the renal sodium and fluid retention, which increases blood volume. There's a lot more stuff that happens with this as well. It affects the pituitary gland. It affects the heart and the vascular system. It affects the thirst response, all these kind of things. But that's the renin, angiotensin, aldosterone system is kind of what we focus on for a lot of the kidney stuff. A little clinical application. You may have heard of ACE inhibitors before. These are antihypertensive medications. This is what they actually do. They actually stop that ACE enzyme from converting the angiotensin 1 into angiotensin 2 to prevent blood pressure from being any higher. Okay, and that is all on our nephrons. We're going to switch gears a little bit and talk about the path of the fluid, so kind of tying it all together, and then also the external passages, so the actual, what path does the urine take to leave the body to be excreted. So we filter the blood in the glomerulus, and it enters the capsular space. It then goes through the proximal tubule, then into the loop of Henle, the descending limb, then the thin, then the thick ascending limbs. Ascending limb of loop of Henle, limb of loop of Henle. Then we go through the distal convoluted tubule, through the collecting tubules or the ducts. The collecting ducts all come together to form papillary ducts. Papillary ducts come together to form minor calyces. Minor calyces come together to form major calyces. Major calyces come together to form the renal pelvis. The renal pelvis drains into the ureter, which goes out of the hilum of the kidney. Ureters take the urine to the bladder, and then finally it is excreted through the urethra. That is the whole path of fluid. Very simplified, but yeah, that's the whole path. So we're going to focus a little bit on those major and minor calyces, the renal pelvis, ureters, bladder, and urethra. Mostly these last three are the main ones that we're going to focus on, but adding a little bit more of the histology information on the major, minor calyces and the renal pelvis. So basic structure of all of this stuff is we have our mucosa layer, no submucosa layer, and then we have a muscular layer. All of the system, all these external passages, so from the major minor calyces to the bladder, everything except for the urethra has transitional epithelium. That's what this is, where it looks very different, you know. It's also called urothelium. If you ever see that in an exam, that's what that's referring to. The mucosa level layer also has lamina propria, but no muscularis mucosa. There is no submucosa. And then we have smooth muscle, where we have areas with inner circular and outer longitudinal smooth muscle. Specifically in the bladder, we have what are called dome cells. This is a type of transitional epithelium. These are special. These are almost always asked about on exams. They ask about dome cells, because this is one of the only places in the body that you're going to see dome cells. So the bladder is originally really small, and then as it fills, so it has the outermost layer of epithelial cells. They look like little domes. As the bladder fills, it stretches, and the domes go flat. And then once the bladder empties, they turn back into dome-shaped. So it just makes something that makes the bladder very unique. There are also 3 layers of smooth muscle in the bladder. We call it the detrusor muscle overall. So if you see that word talking about muscle in the bladder, we call it the detrusor muscle, which is really 3 layers of muscle, the inner longitudinal, middle circular, and then outer longitudinal layers. So here we have our smooth muscle here being the detrusor muscle, and then all of our transitional epithelium in here. You can kind of look and see, if you zoom in, you can kind of see how it's dome-ish shaped like this. Call it dome-shaped, and then when the bladder fills, it gets stretched out. The urethra, we have male and female urethra. The male urethra has 3 specific parts. There's the prostatic urethra, which has transitional epithelium still. The membranous urethra, which this now changes to stratified or pseudostratified columnar epithelium. And then finally the penile urethra, which has pseudostratified columnar with areas of stratified squamous non-keratinized epithelium. The female is much more simple, much more straightforward, I should say, where they just have stratified squamous non-keratinized epithelium with some areas of pseudostratified columnar epithelium. It's important to note the male urethra is much, much longer than the female urethra. If I remember, I think the female urethra is about 4 inches long, and the male urethra is almost double that. Sometimes this comes up as a test question. Yeah, even smaller than that. I was thinking inches, but it's centimeters. So the male urethra is 16 to 22 centimeters long, and the female urethra is an average of 3 to 5 centimeters long. This is one of the reasons why women are much more prone to urinary tract infections as well as bladder infections, because their urethra is there's a much shorter distance from the urethra to the bladder, so it's much more likely that that they can have urinary tract infections as well as bladder infections. Here's what the ureter looks like, the different layers. The male urethra, some of the male urethra structures. There's the male urethra, the penile urethra right there. We'll look at all the male anatomy stuff in the next couple of lectures. This is the female urethra, and another picture of the female urethra. This picture is almost always on tests. This one, I remember this one consistently is tested on. I remember seeing this specific one, I think, 2 or 3 times on different exams. And there we go, the urinary system. So thank you so much for watching, if you made it through all the way to the end. If you have any questions, comments, concerns, drop them down below in the comment section, and I will see you on the next one. Have a wonderful day and good luck studying.